think I do like a lot of different things, but most of my work is I do on a computer. So I think I can do it very well. And I'm going to talk about a lot of different things. I know I have done work on single selling. That's why this kind of work. I'm not going to talk about that today, but yeah, happy to talk about that if people are interested. But I'm going to instead talk about what we've kind of learned from looking at the evolution of sleep. Across different species. So get this over. So I think the biggest challenge for animals on the planet is not like these extreme environments, but it's really like the day-night cycle. So you have these incredible changes in temperature between. So what other animals and things are active change very rapidly between the day and the night. And so most species on Earth have adapted to this in some way. And the main adaptation that I'm really interested in is sleep. So this is where, animals are asleep for part of the day and they're awake for the other part of the day. So in us, for animals, we're awake for the day and sleep at night. But like us, basically every other animal also sleeps or has a sleep-like state. But sleep is super diverse across species. So just like the species that sleep itself is very diverse. So you have animals that sleep at different times. You have diurnal animals that sleep during the nighttime, like us. You have nocturnal animals that sleep during the day, at night. You have species that are active at very particular times. So crepuscular species, when they're active during the dawn or dusk and are kind of inactive in the other periods. And then you also have some species that adapt this by being arrhythmic, right? So they don't really care. So they're active all different times. You also have big differences in the total amount of sleep and the total amount of activity that different species occupy. So large herbivorous mammals like horses and giraffes that are sleep for maybe 2 or 3 hours a day. You have some species of bats that can sleep for up to 20 hours a day. Humans, of course, we average something like 8 hours, but probably everyone in this room sleeps a different amount. So you also have variation within species too, right? This is also quite important and probably also very adaptive, right? To have early birds and night owls. So people around model organisms, this diversity also exists in model organisms, right? So we have mites, which are nocturnal. The other model organisms shown here are mostly diurnal. You also have differences in the amount of sleep. So mites and flies sleep quite a bit, but this is because they have a very consistent nap. So mites will nap at the middle of the night. Male flies will nap in the middle of the day. So you get this difference here as well. And the questions that I'm really interested in are sort of why. So why do we have these differences across species? And then also, I'm trained as a geneticist and also cell biologist. So I'm really interested in the genes and the neurobiological mechanisms that control its variation as well. So there's lots of theories for why we sleep, why animals sleep, when and where and how they do. So one of the big ones is sort of this inactivity theory, where you're inactive and you sleep at a period that you're otherwise not adapted for, right? So maybe it's too dangerous during the day for these nocturnal rodents because of predation predator pressure, so they're better off sleeping in a world, right, and being inactive. There's also this idea of energy conservation. So if you're not doing anything, you might as well conserve your energy. And this is particularly the case when these large predators, right, that eat very frequently, and so they have to conserve energy. There's this idea that maybe it evolved to be sort of a restorative part, right? So you sleep so that you can recover and repair your tissues. And then more recently, it's been kind of observed or hypothesized that it might also have to do with brain plasticity and synapse rewiring, things like this, that those processes happen when you're sleeping and not when you're awake. I think probably all of these are every part true, right? For whatever reason it evolved in the first place, I think the other ones kind of can come after that, right? Because once you have this inactive sleep period, you might as well do some of these other things, right? In general, species tend to be quite adapted to when they're active. And so this is sort of this idea that you might have heard about of the mammalian nocturnal bottleneck. So it's very clear from even our anatomy and our biology that our mammalian ancestors were nocturnal. So the structure of our eyes, but also the fact that we're warm-blooded is thought to be adaptations to being nocturnal, to being active at nighttime. And so there's this idea that the common ancestor of all mammals was nocturnal. This is also not surprising because most mammals are nocturnal in general. But it was recently kind of really well demonstrated through the ancestral reconstruction. And so what this is, and this is a little bit, I do a lot of these, it's on that a little bit more, but here is when you take what's going on at the tips. So here at the tips are all the different extant mammal species, and for the species that exist, so then you know whether or not they're diurnal or not, or at least we can measure it. And then you use this information as well as the relationship in the tree to determine what the most likely state at each of these internal nodes was, right? And so there's some data statistical model to do this. But what they found was basically that up until the KBG, so the boundary between the Cretaceous and the Paleogene, so this is when the meteorite hit and killed all the dinosaurs. Up until this point, all mammals were nocturnal. After this point is when you start seeing the emergence of diurnal species. That is a showing your lineage times one. And the idea is that with the dinosaurs, they were mostly diurnal. They died out. They opened up these niches for mammals to then occupy. So they went into those diurnal niches that were leading by the extinction of the dinosaurs. That kind of the there. Around the same time, a similar study was done, but across all tetrapods. So tetrapods are terrestrial vertebrates, basically. So I'm using like 4 arms, like 4 limbs. So they include amphibians, mammals, reptiles, and birds. And this study did a very similar thing, but across a much larger group. And they found also that the animals were exceptionally nocturnal. But interestingly, they found that all of these figures also exceptionally nocturnal, right? So it seems that, back in the side, being nocturnal seemed to be where it was at, right? All of those species that we know were nocturnal, right? And then they also found that similar to sort of mammals, that when you did transition into diurnality, so in some of these more recent common ancestors, some of the reptile groups and birds, that this was really associated with a lot of evolutionary success, right? So you have many bird species, and that's associated with the fact that on this tree, the birds have more species. So the diurnal lineages here have more species than the nocturnal lineage. Basically, how it's checked. But what I found really interesting about this was the fact that this bird tetrapod, the thing at the back, at the base here, and for hundreds of millions of years, all the vertebrates were nocturnal, right? And so this is kind of interesting because, there was a day and a night back then. So what was going on? Were there diurnal species back then? What happened to them? Is this really true in terms of this nocturnality? And so it's very interesting in these kinds of questions. I think one thing to keep in mind is that tetrapods are a substantive vertebrate, right? So this is the tree, the phylogenetic reconstruction of the tetrapod groups. But if you want to consider what's going on in tetrapods, you should probably consider the other half of the vertebrate tree, which is fish. So fish, particularly bony fish, represent half of all vertebrate species, right? So they are the most diverse vertebrate group. So they are like us. They're very similar to us, except they're really diverse and they live in the water. And this is where this idea comes from that I've heard about fish, right? So we are fish because we are nested within the fish tree, right? So we're not separate. That's a third group. So there's, shooting on this. That's these guys. There. So this is this idea that if we're really interested in what's going on in vertebrates, then we should consider what's going on. Right. However, we don't know. No one knows what the activity patterns of fish are, right? So there are these nice data sets for tetrapods that have been, collected for lots of different reasons, but no one knows what's going on in fish. And so this is, I think, for a couple reasons, some historic and some just kind of mechanistic. So one thing is that fish are super diverse, like I mentioned. So there's many more species. So if you want to get a handle on fish, it becomes difficult because of the number. But also there's a lot of good reasons why you might expect them not to have strong activity patterns. So one thing is that light doesn't really penetrate in water, right? So fish live in water. And so fish live particularly in deep water. There's not a lot of light that penetrates down there. And so the difference between day and night in terms of light, but also temperature is probably not that big, right? So water is also very good buffer for temperatures, right? So maybe the abiotic factors aren't so strong in a lot of fish species or the environments that fish occupy. And so when I tell people that I work on the evolution of fish, right, I always get this question, do fish actually sleep? And of course, the answer is yes, they do sleep, right? There's lots of work in zebrafish that shows that they sleep and that the mechanisms of sleep are very similar to other vertebrates, particularly the way we sleep, a lot of the same circuits and genes and these control sleep in the same way. And so I think that happens. And also, if you ever fish or know a fisherman or anything like this, it's very clear that fish are able to be caught at different times of day, right? Different species. So there does seem to be some activity patterns there. There's some lots of known information. And so this is where we get into this idea of, well, the way to figure this out is we decided to do this extensive literature search, right? So this was mostly at the time manual. It was a little bit aided by some computational methods. Now we're working on new methods where we're like really, it's much more computational. So I have an undergrad here that's working on this and some other things I could talk about it at the end. But for this, it was really all of these manual searches. And I like to call it was just thousands of Google searches. And so this was really, it was quite time consuming and tedious, but a little bit biology. A lot of the data comes from studies like this one in Mola Mola, where they have these like satellite tags to these large fish. So these are these ocean sunfish. They're really big, really weird shapes. But from this satellite, you can really extract like the swimming speed. So that's shown here, and you see these very clear patterns where the swimming speed is higher during the day, and this is associated with these foraging diets. So these species are very clearly diurnal. They don't really do much at night. It's very well known that fish that live on the reefs also have very strong activity patterns, where there's really like species that are active at night and species that are active during the day. They tend to sleep in the same holes in the reefs. They go back to the same place every night to sleep or every morning to sleep. There's even these like parrotfish that blow these like mucus bubbles at nighttime when they sleep. There's really there maybe protection from the isopods and things like that. There's a lot of fish that sing at night. So these are the midshipman fish that have this courtship song. They only sing at night. You can record it in the lab, as shown here, but you can also go out into like a river or a lake and record lots of fish sounds at nighttime. Most fish that sing at night, probably because they use it to find each other right in the dark. And then there's also some examples of fish that these flatfish and these stickleback that if they get infected by parasites, it can actually affect their activity patterns, right? So these stickleback, and particularly in the stickleback, they become active during periods when they're normally inactive, and that increases the ability for birds to eat them. And the birds are the next host for the parasites. And there's a study here that's sort of linked to this. So there's lots of really cool stuff. We ended up getting information for about 6,000 species. It's from 2,600 literature sources. So I read at least 2,600 abstracts. Also read a lot of papers. There's lots of unpublished in here. So make sure you put your data in your thesis, make it in a nice, accessible format. A lot of this is also really old. There's papers from 100 years ago that are like, not even computer readable and things like this. But it's really good to do this. And so now we have this database, right? So we have information for 6,000 species. So now we can start considering doing things like they did in the Patrick model. And so the way we do this is that we need a couple of things. And I wanted to just highlight this because these are some of the bioinformatic methods that I use. So I'll write in that other order. I'll do this first. So first, you need a tree. So we need to know the relationships between all the species in our data set. And so there's a couple of resources that we can use for this. One is this Tree of Life project, but also the Tiny Tree. And the way we can do this is like, what this does is it combines trees from different sources, and you can use like mutual information from the trees to combine them together to make sort of synthetic tree. And then we also have a lot of information from the time calibration. So the idea here is that for ancestral node, if there's a fossil that represents that species, we know how old the fossil is, and so we have some sense of when that occurred, right? And so this is very important if you want to model the evolution, because different species will have different branch lengths, and that's really important. The next thing we do is usually we try to model the evolution of this behavior across time. And for this, it's a discrete trait, right? So we have species that are either diurnal or nocturnal, or crepuscular or ephemeral, meaning it says crepuscular. And so what we do is we actually use these like sort of machine models. So this is the base model, and what we're trying to determine is the parameters that we're trying to optimize for are the transition rates between the densities, right? And so the idea here is that we start at the tips with this information. And so, if you're diurnal, we try to determine what the rate of transition could go to nocturnal. muscular, all of the combinations there. And the idea is to try to optimize those rates so that it's most likely to give us the excellent information during the tree, right? In addition, we do that's number. We had so these are very simply, if you have like these three states that are measurable, right? You have these measurable observable states. You can complicate this model more, for example, by including hidden states. And the idea here is that, for example, if you look at this string, these diurnal species over here versus these diurnal species over here, it's very different, right? These species stay diurnal for a long time. They started diurnal back here and they stay diurnal, whereas these diurnal species are arising from nocturnal to common ancestors. And so the idea here is that maybe this diurnality is a different flavor of this diurnality. And including it in rates allows you to test it. Okay. So what do we get? So I skip a little ahead. So what I'm going to show you here is the ancestor reconstruction now diurnality and nocturnality across 4,000 species. So think of a slightly older analysis here. But we see some very interesting things, mainly two things. One, we see that the common ancestors out of bony fish and of cartilaginous fish, so sharks and rays and things like this, they're both nocturnal. So this is similar to what was observed in tetrapods, except differently from tetrapods, we see a lot more variability on this tree. So there's a lot of diurnal and nocturnal lineages. It's not the same where, for example, all mammals mostly are nocturnal, all birds are diurnal. In fact, like in fish, it's about 50. Right. And we see many unit transitions. So like all of these red lines here, almost all of them represent independent transitions into diurnality. And then you have, for example, this big group here, where the common ancestor probably transitioning to diurnality, and you have many lineages that have transitioned back into nocturnality. Right. So you have this sort of incredible variation here, and that's very different than tetrapods. Looking at the post-combiner data with tetrapod data, and what this does is we see, not surprisingly, that the common ancestor of all vertebrates was likely nocturnal, and we see these differences between clades. We can quantify the number of transitions, and so this is cartilaginous fish and bony fish, and you can see that they have many more transitions compared with other groups. Sauropsids are reptiles and birds that is from here. They also have a lot of transitions, but they're all mostly unidirectional. So they go from ancestral nocturnality to diurnality, and then they stay there. They don't go back to nocturnality. And it's unlike fish. So we do see this very big difference. Yeah. Okay, so I mentioned that the transition in mammals, right, the transition to the diurnality in mammals occurs only after the meteorite killed the dinosaurs, right? And so when we ask, if we have so many transitions in the other groups, we'd ask also when are they happening over like paleontological time limits. And so for this, you're seeing here, so this is sort of classic timeline. You've got this is present day, this is Cretaceous, this is when the meteorite hits. You have these other very, meteorites here. And what I'm going to show you is the sort of the normalized number of transitions relative to the size and relative to the number of species that are happening there. And I'm splitting it now by the major vertebrate groups. And so with mammals, we see the same thing that was previously reported. So we see this peak in transitions after the Cretaceous at the beginning of the Paleogene that correspond with what was seen before, so probably this dinosaur extinction event. Interestingly, we see a similar pattern in amphibians. So we see also a peak after the Cretaceous. We see a peak before a little bit, but I don't know what's going on here. But it seems that maybe this is related, right? Maybe it's the same thing going on. Maybe the dinosaurs dying also opened up niches for amphibians. On the other two groups, we see something similar, but at a different time. So we also see peaks in transitions after a period, but in this case, it's after the Jurassic, right? So before at the beginning of the Cretaceous. And so what we think is that there's something, happening at these transition points that's associated with the ability of these lineages to transition, right? And so our best guess right now is that typically these are defined by periods of increased extinction, right? So a lot of species dying and going out and going away. And so what we think is that the final species rates are really associated with sort of providing the ability for transition into diurnal for the first time. And so basically what happens during extinctions is that you have species that occupied all of the sort of known ecological niches during extinction, and many of them dies. But then you have, after that, you have recovery, and this recovery takes the form of the surviving lineages, diversifying and new species to occupy all of the same ecological niches that existed before, or new ones that were created. And so this is, however, it's long been sort of recognized that these extinction events are not random, but they're very selective, right? So they're killing certain lineages and then allowing other certain lineages to survive and thrive afterward, right? So for example, at this extinction event, right, Ordovician, you had trilobites and brachiopod monogates, and then afterward you had the emergence of fishes for the first time, right? And so these very selective things occurred to it's really unclear whether or not there's any sort of general mechanisms happening at this time points. And what we sort of propose is that maybe the general mechanism is that nocturnal species, for whatever reason, have some better ability to survive these extinction events, right? Because we see, for example, that only nocturnal tetrapods survive some extinction event, and only nocturnal fish survive the extinction event. Is that those lineages that can transition into those empty niches, into those empty diurnal niches, are those that maybe are proliferating in the aftermath, right? And so this is really kind of demonstrated by fish. So we think fish are quite evolutionarily robust, they survive a long time, they're very diverse. And you think that this is really partly associated with their ability to transition. And so what this graph is showing is the number of lineages that have either never transitioned once, twice, 3, 4, 5 times. And then today, only about 5 or 10% of species of fish like never transition, right? So most transitioned at least one. And so this indicates that, those that we have so many species as they've been able to transition. Okay. So this is very kind of interesting, but it's also really out there. I don't have a background in this. I'm not a paleontologist framing on this. And it's kind of hard because you can come up with these great, fantastic hypotheses, but it's very difficult to test, right? You can't test this. You can't do it at extinction, right? But on this side, we can sort of at least look at other cases where this might be happening to sort of see whether or not this is a case in denial. So we have some patients in the aftermath of these extinction events, all that it's need to diversify, right? So the more seed to evolve. And so are there other cases where this is happening that we can see whether or not this mechanism is involved? And the idea there is that there are these other examples, and that's in what are called these sort of adaptive gradations. And so an adaptive gradation, and these are just examples of different adaptive gradations across the world, are in cases where a single lineage, so a single species, comes to occupy a new environment, and then in that new environment, it's maybe newly created or some newly formed. And what it does is that species evolves to become many species to occupy all of the ecological niches in that new environment. And the ones that I work on are cichlid fishes. So here from Africa, that's Dustin mentioned. And so these cichlids are this example close to where I find a really relatively short amount of time. And they've done so in these African-rich lakes. And it's bigger lakes that have been generated in the last 250 to 10 million years because the eastern part of Africa is separated from the rest of Africa, right? So it's making these big holes in the ground that get filled up with water. It's a new lake. Fish from the rivers still eat those lakes. And what happens is that fish with a cichlid and 250 years, 200,000 years later, 10 million years later, you have hundreds of species of cichlids that come from this one system. And what's really cool is that you have these convergent parallel gradations that are happening in the three lakes, and you get the same ecological adaptation. I study fish from Lake Tanganyika, so this is the oldest of the three lakes. So 10 million years old, there's probably 240, 250 species in this lake. And it's been really clearly shown that these species have evolved in a very stepwise manner. So first you have diversification where you have the, as that's what you see, that's two species, one that is occupies the rocky areas, the shores out of Africa, and one that is on the sandy substrate further up, right? Then you have within each of these habitats, you have diversification by feeding apparatus. And so this is really the morphological changes to their jaws. They actually have two separate jaws on an outer one and the inner one. And you have adaptations, yeah, define, say, the algae, you have cichlids and phytoplankton, you have cichlids that eat the scales off of other cichlids, so they really specialize in just eating the scales off of fish. And you have these incredible adaptations there. And then within each of these, again, you diversify by coloration. And so you red one. And so the idea is that you buy two other species because you have one through all of the combinations, right? All of the possible combinations. And the idea here is that you have this explosive adaptive variation, and one way that you could get even more species, right? You could double it, not triple its number, is if you made, diurnal, nocturnal, and crepuscular versions of all of these, right? And so you could ask, as this symbol define a technical term for this, contributed to this diversity. It's an technical species evidence define, and then define information in genomic resources for these species. And so if we do identify some of these differences, we can also potentially get to the generics as well and using these models. Okay, so I didn't sort of define those like that. We collaborated with define like Saltberger, who's really kind of next. So if you're interested in these fish or more information, you should go Walter. So there's 250 species in the lake. Nothing was known about any of them in terms of activity patterns, right? And so what we decided to do was basically, when I moved to Switzerland, the lab, Walter was there, and in his basement, he had 40 species, right? And so we thought, well, we could test them. We could look to see when they're active and when they sleep and all of the different things. And so that's exactly what we did. We ended up doing 60 species from the radiation. We tried to sort of, get species from all around the tree to be representative of the radiation. We also had, for example, the biggest fish in the lake, but also the smallest fish in the lake. So these guys live in these empty snail shells. These are the big piscivorous fish that eat those fish, right? We had fish that represent a more pelagic lifestyle. So these new big schools, they're out in the middle of the lake, right? Not near the shore. But we also have these more basic species. So these are like goby-like. So they live in like the rock crevices and they pop up and they go back down. Like this. We also have these species, Lobogeloides, which have these hypertrophic lips, really big fat lips, and they use these actually to like suck crustaceans out of crevices and rocks and things like this. And then if you know anything about cichlids, you might have heard about Dactytilapia bertoni, and these are sort of one more classic model species from cichlids. And they are riverine species, so they live in the rivers, and they're kind of thought to maybe represent like the more ancestral. Okay. So the way we do this is it's in the lab. So we actually record these fish. So we work with the fish in the lab, and we develop this sort of rig where we could record, hundreds of individuals for a long period of time. So this is the raw video, and this is sort of a processed video, and then we clean the fish. And we do this for 6 days and 6 nights. So every individual will record for time. There's a 12-hour flow of light. But we also sort of mimic dawn and dusk, which is important. And then I could take a look at our breathing here. These are example videos from each of the 60 species. So you can see that there's a lot of diversity both in terms of size, but also behavior, where they are in the tank. We have some species, for example, these ones here, that build these sand manifolds. So they pile the sand every day, and they tear it down, they pile it back up, they tear it down. These are kind of breeding territories they use, right? So they build these things and then they do portrait displays, all sorts of interesting stuff. So we do something very simple. We're starting to get a little bit more complicated now, but for the beginning, we just really track, their speed, their movements, how fast they're moving, right? And this is the water coast, the big cichlid. And you can see that they mostly are swimming much faster during the day than the night. Like blue is the night, the white is the day. So their swimming speed much higher. And in fact, like at the nighttime, these fish are mostly just like, at the bottom of the tank, chilling, not doing nothing, probably sleeping. You can average this across days, so you get this defines this type of define that mola. It's very dynamic, right? So it's very active during the day, not very active at nighttime. We have, in fact, examples of all of the different activities in our fish. So we have, this is another diarrhea species, Yaba with butyride. This is the closely related species that is has the opposite nocturnal. So it's really active at nighttime, very active time of day. These fish have giant eyes, and they actually have this sort of extensive sensory system that also kind of makes sense of their nocturnal lifestyles. We have crepuscular fish. So these are fish that have these peaks of activity during the dawn and the dusk. We even have silent species, the one species that peaks like an hour before the lights come on. They kind of know internally that the lights are going to come on. And then we have some evidence of arrhythmic or ephemeral species. Wow, like don't really have consistent patterns. Since we're measuring this ourselves, we get more information from this, so we can actually extract what we need to measure sleep. So we call it rest in this case. But this is sort of the number of hours per day that we think the fish are sleeping. And we see some species that sleep for only a couple hours, including this one on the mountain, like which only sleep for 2 hours, and they only do it during the day. This species also sleep for a similar amount of time, but they start equally between the day and night. Right, so we see all of the different possible combinations, right? So diurnal, nocturnal, when they sleep, when they are awake. We have ones that sleep a lot. These are crepuscular species, right? But they're really only active during the dawn and dusk, and otherwise they're inactive. If you look across all the species, these are just these sort of big plots. And what you see is just like very, like it's super different. You have all the sort of different possible combinations that you could think of, which is really exciting in terms of like studying this. But sort of these classic definitions are a little bit qualitative, right? So one diurnal or nocturnal. And the way we can actually quantify this a little bit more precisely, right? And so the way we do this is very simple, like principle. So we're quite it's actually how well this kind of simple analysis explains the data, where the idea here is that we're looking for different combinations of the time points that best explain the variability between species. And so the idea here is that PCD1 is really describing the difference in activity between negative time points, your blue and day time points, right? And PCD2 is describing the difference between those around the dawn and dusk, so 7 o'clock in the morning, 7 o'clock in the night, 7:36, 8, those time points at the rest of the day. And so you get these nice plots where this is the contribution of each time points, each critical component. And so PC1, species that have a high PC1 score are nocturnal. They have an activity pattern of studies. Species that have a high PC2 component are crepuscular. They have an activity pattern of studies. Okay. One important thing that we get from this already is that it's kind of thought that diurnality, nocturnality, and crepuscularity were mutually exclusive, right? You're crepuscular or nocturnal. And in this case, I would see it's actually you can be both. You can be both nocturnal and crepuscular, or diurnal and crepuscular, right? You can have high PC1 and high PC2. So it seems like they're kind of independent. And we also have some supporting information from like some species causes, like genetics, where it's even like the morning peak and evening peak are regulated by different genetic and are good. Okay, so I'm going to go a little bit quickly through this, but one way that we can kind of show that we have this nocturnal partitioning and this kind of thing going on is that we have in our data set species from different ecological niches, right? So algivores, vertebrates, piscivores, elanchivores. And one idea that the hypothesis would be is that if there is temporal niche partitioning, then we should see both diurnal and nocturnal algivores, we should see diurnal and nocturnal invertebrates, et cetera, right? And so we do see this. So if we look at the values, right, metrics, we can see that we have the algivores that have low PC1 scores, and algivores that have high PC1 scores, and we have sort of differentiating between. And this is sort of another example to just kind of show you. So these are all the large piscivorous fish that have a lake-wide distribution. We have a diurnal, nocturnal, and crepuscular ones. The crepuscular one is specifically active during the morning. And so from this, I would actually hypothesize there's probably another large piscivorous fish that's only active in right the atopy. So that would be something. I don't know. So we think that it's really contributing. So the last thing, I don't know, can go very quickly through this is that, I kind of promised that we can identify genes. So I also do a lot of genomics and genome-wide association studies and things like this. So we can take this data, right? And if you take these PCT scores, the total amount of sleep or rest that we have, right? And we can actually do a GWAS, right? So it's not exactly the GWAS. So if you, and then whoever does the GWAS, you know that you can really only do GWAS on populations that are intermixing. These are different species, so they're not intermixing populations. Although we think that there's been a lot of hybridization, so we think we do it. The way we get around this is we do it kind of and skip this. Is we do a kind of hybrid approach where we do both a regular GWAS, so this is just a linear association between each SNP and the phenotype, but then we also do a phylogenetically corrected one. And what we look for is, each of these dots is a SNP. And so ideally, we want SNPs that are highly significant by both sexes, or at least one, right? And so if you look at this kind of plot, you can imagine that this is a SNP that we're interested in, that is very significant by both sexes. And if we look at the association between allele frequency and the behavior, we see that all of the species that have the reference allele, they're mostly diurnal. And the species that have the alternative along the other end are all nocturnal, right? And what's really cool is the rapid allele is different species that I didn't test, but other papers have demonstrated that it's diurnal. So we really think that it's kind of the relationship that you expect, right? You know, this nice association. And so, of course, the next question is, where is the SNP? What it's doing? It's actually not in a gene, but it's nearby a gene, probably in a regulatory region, a gene called CRCP, which is a receptor for a neuropeptide called CRDD. And if you have any interest in sleep or brain neurobiology or anything like this, the fact that this is a neuropeptide is really kind of exciting. These neuropeptides are really known to control these kind of brain-wide states, although this one I wasn't familiar with. However, previous work has really shown actually in both flies and zebrafish that it controls wakefulness. So in zebrafish that are missing the pathfinder, they are less awake during the day and they sleep more at night. And flies the same, so they sleep more during both the day and the night. And so the hypothesis here is that potentially the SNP is regulating in the hand, so that controls the expression, and that you can probably have this gene expressed during the day, the diurnal species, and then right. You'd have to link to it and back. Okay. The other little bit of information that I want to share is that originally our kind of hypothesis that we had was that one way you could change from diurnal to nocturnal is by changing the circadian clock, right? So this is this endogenous kind of thing that's going on in your cells where you have these transcription factors that are oscillating throughout the circadian time point because of the effect of these other two genes that they are, right? So you have this circle. But what we find is that we don't think that there's any SNPs that are anywhere near these genes that are associated with the differences. So that's actually shown here where these are any SNP anywhere near one of these genes, and they're all not associated at all with the difference between species and behavior. However, we know that the diurnal and nocturnal patterns are circadianly controlled in these animals, right? So we have these classical experiments, these experiments kind of removes exogenous cues, and the diurnal behavior continues in the absence of light cues, and the nocturnal behavior continues in the absence of light cues. So they're still more active at night than they are during the day, even if the light comes on. And so we think the clock is the same between these animals. And so that means that you basically have to change everything downstream of it, right? You can imagine that if you just, inverted this, everything would come with it, it would all work very well. But instead, what happens is the clock stays the same, but between diurnal and nocturnal species, all of the downstream things need to change. And this becomes really complicated because there's lots of different pathways and circuits and neuropeptides and genes that control these behaviors. And so you can imagine that all of these sort of have to be reprogrammed in some way. So that's the best. But it also means yeah. So just to sum up, this is sort of that we see these kind of similar things happening here, but that really actually studies that are interested in looking at the activity patterns that fish are really a place to look at. You have so much diversity, and you have really kind of a lot of transition and a lot of evolution happening here in terms of. So gives you a lot of power to do some of these analyses. Yeah. So just to sort of one thing that I don't know, and we'll give a feedback for is we don't really have a good explanation for why nocturnal species survive in extinction events. So we proposed something in the paper, the reviewers didn't like it. And what we proposed is we thought it had to do with temperature, but that doesn't seem to be really a clear thing. So if you have any other ideas, I'm happy to hear it. Um, we're also now, I sort of got into this, we're thinking about what about other animal groups, I think particularly insects, right? So can we look at this in insects? And that's where we're getting a bit more sophisticated with some of our analyses here. We're looking at these big databases more computationally, and we're also doing, different kind of generally different, and like random forest models and kind of simple things to help prioritize some of the literature on this, and that's what Peter's working on. Um, and then we're looking more now into the neurobiology. You do the Sigma now as a model, right? We'll be background on monkish, doing functional genomics, but also a lot more behavioral experiments. So just to wrap up with the acknowledgements. So I did my postdoc at Alan Shear's lab, but we collaborated with the lab called Pluggers. There was a lot of people there that I worked with. We did a little bit of field work that I didn't talk about. So we collaborated with the University of Zambia and also the Ministry of Fisheries in Zambia. And then I have like a small lab now, where we're slowly growing. Happy to take questions.